This is the Professor, C.B. M.D. Ph.D. and today is Tuesday, December 3rd, 2024. It is 8:54 and 13 seconds, p.m. Eastern Standard Time, and today's research, exocytosis. So let's hop to it here. The movement of secretory vessels to the plasma membrane is essential for normal cell function. The fusion of secretory vessels with the plasma membrane is a complex process and involves various types of proteins and lipids. Understanding the underlying molecular mechanisms of exocytosis and membrane fusion provides a solid foundation for pharmacologic treatment of many diseases. A variety of molecules produced by the cell for export are initially delivered from the site of their formation of the Golgi apparatus. The next step involves sorting and packaging the secretory product into transport vessels that are destined to fuse with the plasma membrane in a process known as exocytosis. Intercellular traffic of these vessels is achieved by the presence of specific proteins on their surface, codimers such as COP1 and COP2, that mediate their movements. The molecules that travel their route are often chemically modified, e.g., glycosylated, sulfated, as they pass through different cellular compartments. The membrane that is added to the plasma membrane by exocytosis is recovered into the cytoplasmic compartment by an endocytic process. There are two general pathways of exocytosis. In the constitutive pathway, substances designated for export are continuously delivered in transport vessels to the plasma membrane. Proteins that leave the cell by this process are secreted immediately after their synthesis and exit from the Golgi apparatus, as seen in the secretion of immunoglobulins by plasma cells and of collagen by fibroblasts. This pathway is present to some degree in all cells. The TEM reveals that these cells lack secretory granules. In the regulated secretory pathway, specialized cells such as endocrine exocrine cells and neurons concentrate secretory proteins and transiently store them in secretory vessels within the cytoplasm. In this case, a regulatory event, hormonal or neurostimulus, must be activated for secretion to occur, as in the release of secretory vessels by chief cells of the gastric mucosa and by acinar cells of the pancreas. The signaling stimulus causes a transient influx of Ca2 into the cytoplasm, which in turn stimulates secretory vessels to fuse with the plasma membrane and discharge their contents. In the past, secretory vessels containing inactive precursor zymogen were called zymogen granules. In addition to excretory pathways, proteins can be transported between the Golgi apparatus and other organelles along endosomal pathways. These pathways are used for delivery of organelle-specific proteins, such as lysosomal structural proteins, into the appropriate organelles. And this is Professor Christopher, codename Will, leader of research scientist ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. This is the Professor, C.